So the next topic we have today is various enzymatic activities which are acting on histones. We have seen histones, they are very important constituent of the nucleosomes, and they play a very important role in the compaction. And while compaction, there are various rather we have seen around 5 types of bondings are there, and these bondings which are weaker bonds, and this weaker bonding causes or it allows the wrapping around histones. The DNA is wrapped and it can be whenever required it can be rewound or it can be taken off. So like that it is there, and therefore it plays very important role. And these histones, they have their own tails. In tails there are variety of protein molecules are attached. We call them as a post-translational proteins, and there are various amino acids which can be additionally attached or in the protein. Whatsoever amino acids are there, various molecules can be attached, and because of this attachment it can cause the changes, and this result into changes in the activities. This is what we have seen, and accordingly how the nomenclature that changes. We have seen that example, if it's a lysine amino acid is having any substitution or any molecule which is attached, then we call it as a we will give the number. Suppose if it is histone 3, then we give histone 3, then K for lysine, and at what position it is, like that, and which kind of molecule, whether it is acetylated or something like that. So we have seen this kind of activity, and this has given us indication that they are important in various kind of regulatory activities. So let us see them briefly, although these are very detailed one, but we will make it short. Only what's very important, that much only will go for. So we know what is it is. These are acetyl transferase. They are also called as HATs. They are the enzymes. They acetylate the lysine amino acids on the histone proteins. They by transferring an acetyl group from acetyl CoA to this acetyl lysine, they will make it is an acetyl lysine. We know that DNA is wrapped around histones, and because of the transpiration of the acetyl group, the genes can be turned on and off. Means the expression of gene. What is the expression of gene? Gene expression, this word will come very frequently. So let me tell you, anything that results into the changes, maybe it is example replication which is causing cell division, or formation of RNA which result into protein formation. So anything which causes processing of the genes for any kind of functional activity, may it be cell division or may it be protein synthesis, we call it as a gene expression. So genes can be turned and off, and accordingly the expression turned and off and on, and accordingly this is a very good regulatory mechanism on the genetic expression. Histone acetylation increases the gene expression, and there are enzymes which will cause deacetylation. We shall be seeing them also. HDACs, okay. So these are opposite to this acetylation. They will not do the acetylation, rather they will remove the acetylation. So it is linked to the transcriptional activation. I am telling you there are two ways of expression. Expression is, one is for replication and other is for transcriptional. So it is linked to the transcriptional activity. It is associated with the euchromatin, which is less densely compact, allows transcription factor to bind more easily to the regulatory site on the DNA, and it causes transcriptional activation. When it is first discovered, it was thought that acetylation of lysine neutralizes the positive charge which is present, and because of this the unwinding becomes easy. Okay, and then later it is found that the lysine acetylation and other post-translational modification of the histone generate binding sites for specific protein-protein interaction domains, like acetyl lysine binding bromodomain. Okay, you need not to remember all this name, but this is happening. This was a new formation. Earlier it was only believed that since because of the acetylation is there, so the lysine will be bind more to the acetyl group, and because of that its affinity with the DNA will be less, and because of the less affinity, DNA can be easily accessible for the transcriptional factor on It can be easily unwound, and unwinding will cause easily expression. So this is a multiple way by which this is acting. There are variety of HAT families are there. Basically two different classes based on their subcellular localization. Type A is a family which are located in the nucleus, and they are involved in the regulation of gene expression through acetylation of the nucleosomal histones in context of the chromatin, and they contain a bromodomain. As I told you, bromodomain is the portion that is responsible, or that is important for the expression. It helps them to recognize and bind the acetylated lysine residues on the histone substrate. These are various examples like GNC5 etc. Will not go in more details, we don't want to have that much details. The type B type are there, which are located in the cytoplasm, and they are responsible for the acetylating newly synthesized histones which are prior to their assembly into the nucleosomes. So this is how they are there. This histone acetyl transferases, they are lacking a bromodomain because their targets are unacetylated. So the acetyl group which are added to the type B type of agents, they are removed by this deacetylator enzyme, that is HDACs, that is histamine deacetylators, once they enter the nucleus and they are incorporated into chromatin. So these are there. There are functions in multiple complex. We shall be seeing the functions in details. There are variety of families again we can have. So there are out of that this is one of the family, that is GCN5 related N-acetyl transferases, that is GNATs. Okay, in GNAT again there are many activities, many agents with structural differences. I will not go into more details about it, because again that much detail is not expected we should have. These are responsible for various kind of activities. It is part of the RNA polymerase II holoenzyme, and it plays important role in the transcriptional elongation, that is elongation of the transcriptional RNAs. So it is important into it. Then another family is MyST hats. So this family is named after its four founding members, that is MOZ, Y, S and T. You can see that is MOZ, YFB2, SAS2 and TIP60. So these are there. Again here there are many subfamilies, will not go into that much details, and they are found to acetyl. All these are responsible. So earlier we have seen bromodomain. These are presence here, the important things present which are known as zinc fingers and chromodomains. Zinc fingers, the word will come many a times, because these are the proteins which are actually based, or they are having a complexes with the zinc molecule, and zinc plays very important role into it. And earlier we have seen bromodomain, here they are chromodomain. Chromo is a color, and they are found to acetylate lysine residues on the histones like H2A, H3 and H4. They contain zinc fingers as well as the highly conserved motif A that is found amongst the GNATs that facilitate the acetyl CoA binding. So a cysteine rich region that is located on the N terminus of the HAT domain of these mixed proteins, and they are involved in the zinc binding. It is essential for the HAT activity, that is histone acetyl transferase activity. Now they are important in again various activity. There are some repression region, there are some expression region, and accordingly do the balancing of the repression and the expression. So this nice balance it is maintained by this kind of activities. Then they are also important components of the ORC, that is origin of replication complex. You remember the first stage in the replication was the origin of the receptor replication complex, or we call it as a ORC, and that was the first step, and that is how this plays important role. There are other proteins are also there apart from these two major families. mist family and the previous one gnat family, there are other members are also there, and they found typically in the higher eukaryotes that exhibits HAT activities. Again here there are many such examples are there. Will not go into each and every because it is very complicated otherwise, and it is beyond of our scope. So there are many other families are there which are playing important role. Basically what we should understand that they are playing important role. These all acetylators, simply as we have seen that they will cause the acetylation of the lysine moiety in the post-translational proteins, and because of that they will play important role. A few more important proteins are present to that, what we will call it as a nuclear receptor co-activators. So there are three important nuclear receptor co-activators are there that will display this histone acetyltransferase activity. They are SRC1, ACTR and TIF2. These are various such proteins. The SRC, that means steroid receptor co-activator 1, is known to interact with the other proteins, and they will all this they will act at one or other place, and they will make the what we can say gene expression regulation in a proper manner. So this is how these agents are acting. I will not go into these families and description. Okay, there are so many genes and so many structures are present. So what are the overall structure? Again I will not go because we are not in much interest with various structures which are there. Basically only important thing you can remember it, in the mist type, the another type, there are zinc finger, that means zinc is there, and zinc causes zinc acts, and the finger like protein are there. So they are looking as a they are therefore called as zinc fingers. Okay, so these are various families and various proteins which are there. We shall see only the functions of important functions of these. I will not go into each and every kind of protein which is there. Only you remember these are the name, like P300 CBP family, RTT109 family. Okay, and what are the mechanism they are doing? So there are families, as we have seen, gnat and mist families we have seen. So what activities they are doing? So as we have seen, the job is acetylation, that is they will do the acetylation, and this acetylation is transferred to the lysine side chain, and because of that it will be acetylated. So they are important, and the acetylated histone is then released after that. So they are doing the catalysis of transfer of acetyl group from acetyl CoA to these histone structures or these proteins which are there. Similarly about these various mist family, P300 and RTT109, these are there. These different agents, they have different kind of lysine selectivity, and again it's not that important for us. At least at our level we are not going to be geneticists, and therefore we are not going to go for more details. Okay, so important is this, how this is regulated and what are the activities happening. So catalytic activity of these is regulated by two types of important mechanism. One is interaction with the regulatory protein subunits, and other one is autoacetylation. These are two important activities. This acetyltransferases can be regulated in multiple ways, and the same effect or may actually lead to different outcomes under different conditions. This is very important, that is that acetyltransferase enzyme is regulated in multiple ways, means there can be many molecules which can be either increasing or decreasing, or which can be stimulating or inhibiting it, is one thing. Second thing is, just like heart, if you know, the heart is having so many, there is a SA node, there is a vagal activity. Okay, so multiple regulation is there, and there are many other chemicals which can also be acting on that. So this is how it is there. Then the same effector may actually lead to different outcomes under different conditions. That means it can be different action at different time, and therefore it becomes this is very complex regulation that is maintained, so that if anything has to be maintained, or homeostatic activities or normal physiological activities of this gene expression has to be done, this multiple control serves the purpose. The association of these enzymes with many protein complexes that provides mechanism for the regulation of both HAT activity and substrate specificity in vivo. The molecular basis how it is occur is not known, because it is very complex, as I was telling you. So how it exactly it is happening is not known, or how, at what condition, what type of chemical or regulatory is secreted is not known. We know many things about, example, heart. If suppose baroreceptor, you know chemoreceptor, and because of that there is a sympathetic activity and something like that, or flight and flight mechanism, opposite to that is a parasympathetic activity. So some of the things that in what condition what will be activated, but here it is not exactly clear why which chemical is activated and how it will be regulating, whether it will be down regulating or up regulating or increase decrease, whatsoever it may be. So these are not exactly clear because it is so complex now. Fortunately, we know even up to this is a good thing, which was not known earlier. The association of HATs with the multi-protein complex that provide a mechanism for regulation of both HAT activity and substrate specificity in vivo. The molecular basis is still not known, and the mixed family of this is regulated by autoacetylation. The various types of proteins, they are autoacetylated and they are conserved as active site lysine residue. So this is very important, and they are playing important role. They are always having interaction, or they are always having sort of equilibrium with HDACs, that is histone deacetylase. So acetyl transperases will cause acetylation, and deacetylases will cause removal of the acetyl group, and that is always equilibrium that is maintained, or that is always a balance is maintained. That is acetyl transperases will do one job, and then deacetylases will cause the removal of acetyl group, and thus it will reverse the action of HATs. So these are recruited through their target promoter through a physical interaction with a sequence specific transcription factor. They usually function within a multi-subunit complex in which the other subunits are necessarily for them to modify histone residues around the binding site. The histamine, these enzymes can also modify the non-histone proteins, and because of that they are doing many biological roles. So what are their role? One role is chromatin remodeling, that is the structure of the chromatin is modified in a better position. So they serve for many biological roles inside the cell. You know what chromatin is. Chromatin is a combination of proteins and the DNA which is found in the nucleus, and it undergoes many structural changes at different cellular, like DNA replication, DNA repair, transcription. At every process chromatin has to undergo the structural changes. So if the chromatin doesn't do this, then these things may not be possible properly. So chromatin in the cell can be found in two states, either in condensed or uncondensed. Condensed means they are compressed. Uncondensed, particularly it happens during the replication. We have seen that it enlarges. If you remember, from the normal size it becomes almost 1.5 or up to twice the size increases. So this is there. The uncondensed form is known as a euchromatin, while the transcriptionally active condensed one is called as a heterochromatin. It is transcriptionally active. Histone, we know that there are 4 histones. Out of that 4 are the core one, and one H1 is the linker one. We have seen it last time. They will form the complex. We know that it will form octameric complex. We also know that it will link 147 base pairs and we call it as a nucleosome. So they are important in compression, or what we can say condensation of it. And this positively charged, we have seen various 5 types of charge are there, or 5 types of bonding the histone can have with the DNA. So out of that, one important is there, that is positively charged with the N terminal, and the phosphodiesterase backbone of the DNA is a negative. So this positive and negative charge they will call as the electrostatic force, and because of that a strong ionic interaction between histone protein and the DNA. So this transfer of acetyl group to the lysine will neutralize the positive charge. So thus the interaction will be reduced, and that will be easy to remove then, because this bonding will not be there, the bonding will not remain stronger. So it can be easily uncoiled, and apart from that we have seen that it forms the a particular structure to what we call as a bromo core molecule. So these are the things which are there. Different level of histone acetylation as well as other type of modification that allows the cell to have control over the level of chromatin packing. So if too much packing, then it will be difficult to go for gene expression, and at every level the different packing will be there during replication, transcription, recombination, repair. These are the important processes that are taking place. Acetylation is not only the regulatory one post translation, but to histone, but it also help in other activities also. The combination of different covalent modification of these proteins, that is histone proteins, it has referred to as a histone codes, and it is thought that this code may be heritable and preserved in the next cell generation. So if anything goes wrong with this histone code, can also make problem, or this genetic problems. So this is how this whole system is working. Now we know what is gene expression. We have seen that what is a gene expression, and they are important because there are two ways. One way they are causing as a neutralization of charge, and therefore easy release of DNA is one thing, and they are forming the bromodomain. So there are two important things we have already seen. So they are important in that way. They also act as a transcriptional co-activators for the gene silencer, and they are most often found in the large complexes which are made up to 10 to 20 subunits, and they are shared with the different HT complex. They also includes many other proteins and the activating factors which are playing role, and they all will modulate the histone acetylation acetyl transferase activity by bringing HTs to the target genes, and they can acetylate the nucleosomal histones. Some HT transcriptional co-activator, they contain bromodomain, as we have discussed earlier, and because of that they initiate the activity. Now what is the clinical significance of that? A very simplistic way I can tell you is, example, wherever these activities will be happening one after another, that much portion will be easily can be uncoiled from the histones, and that can be easily then either replicated or can be transcripted. Okay, so it plays very important role in this kind of activity. So that much portion, if it is important for any kind of enzyme or any kind of structural protein, then it will be accordingly it will be transcribed. So let us see what are various activities. So this is to manipulate the chromatin structure, and it gives an epigenetic framework that makes them essential in the cell maintenance and survival. And the process of chromatin remodeling, it involves many enzymes, including these HATs, and we have seen all these enzymes. We have seen the methylation, we have seen the ubiquitination, we have seen the other activity, that phosphorylation. So there are many things are there that are happening. Methionine is attached. So there are many such amino acids are attached. They are implicated as the accessories to disease progression. This is very important. Here why we are studying, this is the basis why we have studied it. So this histone acetyl transperases, they have been implicated as accessories, or they are supporting the disease progression, particularly in neurodegenerative diseases. Example is Huntington's disease. If you know, it is just opposite to myasthenia gravis, if you remember last year's. So this is Huntington disease where acetylcholine secretion is comparatively more, and therefore the person is having a dance like movement. So it is also called as Huntington's chorea, that affects the motor skills and the mental abilities. The only known mutation that has been implicated in this is the protein huntingtin is the protein, and it is reported that this is directly interact with the HATs and represses the catalytic activity of these various subunits or subfamilies of HATs. Then other also is there, that is human premature aging syndrome, that is progeria. If anybody remembers progeria, I'll be happy. सुना है कहीं ने progeria? Anybody knows anything about progeria? Somewhere anybody has heard? Okay, that means nobody. So it is again well known, very famous movie. It was there with Amitabh Bachchan and Abhishek Bachchan together, that was known as a Pa, where Amitabh Bachchan has played the role of son of Abhishek Bachchan, and the child grown older than the father. So that was a very famous movie, good movie, that was known as a Pa, and it was based on the disease that is called as a progeria. So it is caused because of the mutational defect in the processing of the lamin A, which is a nuclear matrix protein, and the recruitment of repair proteins to the sites of the DNA damage size is not there, or it is delayed, and that causes the problems in that, particularly in the histone acetylation defects, and the histone H4 is not properly acetylated, or it is hypoacetylated at lysine 16, that is H4K16. We have seen the nomenclature, and this defect is due to the reduced association between histone acetyl transperases to the nuclear matrix. Then another diseases are there. So there are a variety of these diseases are there which are resulting into that. So therefore it is important, therefore it plays important role in that, and therefore we have studied this histone acetyl transperases. Opposite to that is histone deacetylator, and these are what they will be doing. Just a minute. So these are again the another thing, that is histone deacetylases. They are also abbreviated popularly as HDACs, and as we have seen the role of acetylation, it is reverse of that. It too have many families, it too have super families, and once they are doing this deacetylation, once the acetyl group is removed, then histone will wrap the DNA more tightly, because there are two things, that bromodomain is now not activated is one thing, and second thing is the positive charge which was neutralized because of it, now it is again there. So positive negative interaction will be there, and therefore the histone will wrap the DNA more tightly, and it is because that DNA is wrapped around the histone, and DNA expression is regulated by acetylation and deacetylation. So whenever there is a expression is required, then it should be acetylated, and when it is not required, it should be deacetylated. The action I told you, it is just it is opposite to the histone acetyl transperases, and this is how there is a regulation. Okay. uh, they are also called as a KDAX because we have given the lysine name as a K, so, isn't it, in nomenclature. So HDSs are also called as a lysine deacetylases or K, because it is only amino acid where this acetylation deacetylation takes place. Okay. So these are the agents. So there are variety of families are there. Will not go into detail, but yes, I just want to tell you what are the importance of that. So there are 4 important classes depending on the sequence of the amino acid which is present and what are the functions. So see, you can see that what are the substrates of it: HDAC1, HDAC2, HDAC3, HDAC8, HDAC4, okay, 5. This is 1 up to 8. There is a 1 family. Then the second one is a 2A class. Then there is a 3B class. There is a 3 class and 4 class. Except class 3, they are containing zinc. Therefore, we call it as a zinc finger, I told you earlier. And they are known as a zinc dependent histone deacetylases. They are classical arginase folding and structurally and mechanical. They are fold like structure, like finger like structure, and therefore zinc finger we call. Mechanistically distinct from C-twins. These are the class 3 type of, which are folding into the different classes. Now the thing is, we should know where they are. See, ubiquitous, this is in local, these are various classes, 1, 2, 3. In that members are, various members are there in that. How many sites it can be doing, all are doing on one site except HDAC6 and HDAC11, they can act on 2 sites. Then where they are present, they are nucleus, cytoplasm, which organ particular or which type of tissue. Ubiquitous means everywhere, all places it is present, but some are very specifically present in few tissues only. Example is HDAC4, it is present in heart, skeletal muscle, brain, like that. Okay, so there are such, then HDAC6 in heart, liver, kidney, placenta, HDAC10 liver, spleen, kidney, so it is not present in other tissues. Now there are variety of substrates on which a particular place it will be more activity. So these are the androgen receptor, then various, if you remember JAK-STAT and all, P53 in cancer if you remember. So in all this kind of activity it is there. There are many such receptors are there. Variety of proteins are there. Now what will be happen if this gene is not there? We call it as a knockout. If you remember this word we have used earlier also. Knockout means that particular gene is removed. Okay. So if it is an animal, if it is a mice, and in that this HDAC1 is not there, we will call it as a HDAC1 knockout mice. So in that case what will happen? It will show the embryonic lethal increased histone acetylation, and there is increase in the proteins like PP21 and P27. If it is a knockout of HDAC2, if HDAC2 is not present, so that gives idea that because of that what will happen? It will show the cardiac defects. Okay. And so more. If it is HDAC knockout is there, then vascular integrity will not be properly there. Okay. So thus we understand that how this histone deacetylators, where they are present, and if they are not properly present, then what will be there. There are subtypes are there. We have seen them in a table, and these subclasses are having their own role. We have seen them in a table also. So thus HDAC1, these are playing important role. We have seen in table that at various cell how they are distributed. We have seen some are more, some are present everywhere, that is we call them as ubiquitous. Some are present in the heart cells, kidney cells, liver cell, etc. So accordingly we can have a variety of places where these are present. Now, what are the functions of these? So, they are responsible for modification of histone tails. We know everything now, how this they are acetylated, and because of that what it will be there, how this causes charge or discharge, and because of that what will happen. And there is a decrease in the binding, because if it is covalently bound with the histones, because of the deacetylation the charge will be there, and because of the charge the binding will be more stronger, and if it is stronger then it will not be expressed. So this decreased binding allows chromatin expansion, permitting genetic transcription to take place. Histone deacetylases remove the acetyl group and increases the positive charge, and it prevents the transcription. So this is important activity which they are playing. It is involved a series of pathways in the living system, and like environmental information processing, in that all signal transduction are there. Then cellular process, cell growth and cell death, they are important in that. Cell death, there are various death systems, that is apoptosis and all, we have seen during the various signal transduction pathways. We have seen how these signals are brought, and that is MAPK or JAK-STAT and all, we have seen them, and these pathways how they will bring the signal of death like apoptosis. Then various human diseases like cancers, leukemia, etc. So in that variety of these pathways are important. So we have seen that how these are important, how they are having their role at one or other layer. It can also have non-histone effects. So they can also have effect by particularly post-translational modification. This term again, as I told you, theoretically we can understand post means after, translation means the translation is there, and after that what happens in translation, protein is synthesized. The protein which is synthesized during translation, when it is modified after some time, not during the translation, because whatsoever final structure is there, that is in translation, but after that if it is modified, then we call it as a post-translational modification. And this attachment of acetyl group or methionine group or any other group, it is a post-translational modification, because protein is already formed and then we are attaching one or other group, so we will call it as a post-translational modification. The protein phosphorylation is the most widely studied and understood modification, in which the certain amino acid residues are phosphorylated by the action of protein kinases, and these protein kinases are dephosphorylated by phosphatases. So these are various activities, and some examples we can see of these. So they are responsible for the actions of aggresomes. They are tagged by ubiquitination. This is once this is ubiquitin protein is there, if it is attached to any protein, then it causes the damage of that. So thus they are playing important role in removal of the wrong structures. So thus it is helping in the DNA or other important repair mechanisms. Then another phosphate is there, that is PTEN, and that is signaling via phosphoinositol. If you remember, that is PI3, it is there, IP3, what or we call it as a, so phosphatidylenol triphosphate and other things are there. So they are controlled. Then other is APE, REF1, they are controlled. NF-kappa B we have seen recently in signal transduction. So these are the transcription factor effector molecule in the response of cell stress, and it is also there. So they are such regulation is done, and they are known to be active in various degenerative diseases. There are many such diseases which are there. We have seen just now Huntington is recently we have seen, and other like ataxia, telangiectasia. These are various neurodegenerative diseases are there, in which it is there. and it is known to play role. Now there are certain drugs which are known to have the action on this SDAC inhibitor, and they are available in the market. Example is there valproic acid. Last year you studied it in epilepsy, if you remember. So valproic acid is there. It is studied as a, apart from valproic acid, as in pharmacology book we rarely mention it as a SDAC inhibitor. We rather mention it as a what? The inhibitor of GABA transaminases, isn't it? If you remember, I don't know whether you remember or not. इस साल एग्जाम नहीं हुआ तो फिर याद रखने का काम ही नहीं आया है, नहीं? You are lucky enough. So the valproic acid is known to have this action, and they are studied as a treatment for the neurodegenerative diseases. Other such drug which are inhibition causing this acetylation inhibition is there. Example of these are vorinostat. It was approved in 2006 for the cutaneous T-cell lymphoma. Then other drug is Istodax or romidepsin. It was approved in 2009 for again cutaneous T-cell lymphoma. Now these all will be helpful in your cancer also. This can be asked in cancer also. So these are various new drugs which are available for cancer, which are not normally figure in the classification or the list of drugs which are available for cancer. They are also investigated for the chemosensitizer for the cytotoxic chemotherapy or radiation therapy. Along with the DNA methylation inhibitor. This is how additional way it is thought for the cancer treatment. Then some are also modulate the latency of some viruses resulting in the reactivation, and it is shown to occur in some herpes virus 6. So this is another is there. One more SDAC inhibitor, that is trichostatin A or TCA, that is tried for the this SDAC acting on the non-histone proteins, and particularly the repression of the genetic activity, because it will not allow gene to be get expressed. So if you are not allowing genes to get expressed, then we will call it as a repression. Histone deacetyl inhibitors show activity against certain plasmodium species also, and it may indicate to have potential anti-malaria treatment. So this is again under pipeline or under research. So here we finished histone acetylation and histone deacetylation. They are very important in understanding the various important stages or important regulating mechanisms in the what we call it as a gene expression. So I hope you understood this much, and take care. If you have any difficulties, can definitely put on our WhatsApp group.